Hello and welcome to some biology class. Friends, today we shall discuss about the mechanism of transcription in eukaryotes. Before that, I request all of my friends, please subscribe my YouTube channel. Eukaryotes have three distinct nuclear RNA polymerases, each of which transcribes a different class of RNA. Like bacteria in eukaryotes, transcription also involves a series of well-defined steps that are grouped into three stages: initiation, elongation, and termination. Initiation is the first stage of transcription. It begins with the binding of general transcription factors along with RNA polymerase to a promoter region. This binding results in the formation of a pre-initiation complex. Unlike bacteria, which requires only one additional initiation factor sigma, several initiation factors are required for significant and promoter-specific initiation of transcription in eukaryotes. These initiation factors are called general transcription factors. In vitro, general transcription factors are all that is required together with RNA polymerase to initiate transcription on a DNA template. However, in vivo, the DNA template is incorporated into nucleosomes. Under these circumstances, general transcription factors are not sufficient to promote significant expression. In vivo transcription initiation requires additional factors including the mediator complex, the DNA binding regulatory proteins, and chromatin modifying enzymes. 5 general transcription factors are minimally required for initiation of transcription by RNA polymerase II in reconstituted in vitro system. As already mentioned, initiation is the first stage of transcription. It begins with the binding of general transcription factors along with RNA polymerase to a promoter. The eukaryotic core promoter refers to a minimal set of sequence elements that are required for accurate initiation of transcription. A core promoter is typically about 40 nucleotides long, extending either upstream or downstream of the transcription start site. Core promoter for RNA polymerase II contains 4 sequence elements. This figure illustrates the presence of 4 conserved sequence elements in the core promoter for RNA polymerase II. These elements include transcription factor 2B recognition element BRE, the Tata box, the initiator sequence INR, and the downstream promoter element DPE. These are the consensus sequences of these elements on the non-template strand. A core promoter typically contains only 2 or 3 of these 4 elements. Beyond and typically upstream of the core promoter, there are several other sequences which are necessary for initiation of transcription in vivo. These sequences together constitute the regulatory sequences that include the proximal promoter element, the upstream activator sequences, enhancers and silencers, boundary elements, and insulators. All these DNA elements bind regulatory proteins such as activators or repressors that help or prevent the initiation of transcription. General transcription factors help RNA polymerase bind, promoter, and melt the DNA. They also help the enzyme to escape promoter and enter into the elongation phase of RNA synthesis. Pre-initiation complex is formed at the Tata box. The Tata sequence is recognized by the general transcription factor TF2D. The component of TF2D that bind the Tata box is Tata binding protein TBP. Other components in this complex are called TBP associated factors. Upon binding DNA, TBP extensively disturbs the Tata element. The resulting TBP DNA complex provide a platform for recruiting other general transcription factors and RNA polymerase itself to the promoter. This figure illustrates the mechanism of transcription initiation by RNA polymerase II. It shows the order in which various transcription factors assemble at the promoter to form the pre-initiation complex in vitro. After the binding of TF2D, another factor TF2A also binds to the promoter region. This is followed by the binding of TF2B, after which another factor TF2F together with RNA polymerase II also assemble at the promoter. This is followed by the binding of TF2E and then by TF2H. The complete set of general transcription factors and RNA polymerase together bound at the promoter is called the pre-initiation complex. Formation of pre-initiation complex is followed by melting of promoter DNA. Melting requires the hydrolysis of ATP and is mediated by another factor TF2H. Its helicase-like activity stimulates the localized unwinding of DNA around the transcription start site. This localized unwinding results in the formation of transcription bubble. The transcription bubble is a locally unwound segment of the DNA. Separation of two DNA strands in the transcription bubble enable the template strand to pair with incoming ribonucleotides. Initiation takes place within the transcription bubble. Like replication, transcription also occurs in 5' to 3' direction. But unlike replication, in transcription, only one of two DNA strands is used as a template for RNA synthesis. The strand is called the template strand, whereas the other strand is known as the non-template strand. After localized unwinding of DNA, there is a period of abortive initiation in which short RNA chains are synthesized and released. This abortive synthesis of RNA stops only when the RNA chain has attained a length of 10 or more ribonucleotides. In eukaryotes, escaping the promoter involves the phosphorylation of RNA polymerase. This is not seen in bacterial system. The large subunit of RNA polymerase II contains a carboxyl terminal domain which extends as a tail. The carboxyl terminal domain contains several repeats of heptapeptide sequence. Each RNA polymerase II has 26 of such repeats, whereas in case of humans, 52 repeats are present. Each repeat contains sites for phosphorylation by specific kinase. The form of RNA polymerase which is recruited to the promoter initially contains a largely unphosphorylated tail, but the species which is present in the elongation complex has a number of phosphate groups on its tail. The presence of phosphate groups on the carboxyl terminal domain help the RNA polymerase to shed most of its transcription factor, and then it escapes the promoter and next it enters into the elongation phase of RNA synthesis. In case of eukaryotes, high and regulated level of transcription involves several other factors such as the mediator complex, transcriptional regulatory proteins, and chromatin modifying enzymes in vivo. After shedding most of the transcription factors and escaping the promoter, RNA polymerase shifts into the second phase of transcription, the elongation. For this, the enzyme acquires new factors called elongation factors. There are different classes of elongation factors, some of which increase the overall rate of transcription, others help through transient pausing sites, and remaining helps polymerase to transcribe through chromatin. One of the elongation factor is PTFB, which is particularly important. It phosphorylates the serine 5 of carboxyl terminal domain repeats of bound polymerase II. It also phosphorylates and activates two other factors. One is SPT5 and the other is PATSF1. SPT5 is a universal transcription factor. It helps recruiting the capping enzyme to the polymerase II, which is phosphorylated at serine 5 of its carboxyl terminal domain. Another factor, that is TATSF1, helps RNA polymerase to recruit the splicing machinery to the serine 2 phosphorylated carboxyl terminal domain. In addition, PTFB also suppresses the transient pausing of RNA polymerase at certain sequences following initiation. Early in the elongation phase, the 5' ends of eukaryotic pre-mRNAs are modified by the addition of 7-methylguanosine caps. It occurs when the transcript is about 30 nucleotides long. Transcription elongation along the double-stranded DNA is not a smooth ride. For one or another reason, RNA polymerase stops temporarily at certain sequences during elongation. That means polymerase do not transcribe a gene at a constant pace. Pausing is especially pronounced at nucleosomes. It can also be used for proofreading. Recall that in eukaryotes, the genes present in chromatins are organized into nucleosomes. Now the question is, how does RNA polymerase is able to move past the nucleosomes? Surprisingly, this enzyme is able to move past nucleosomes with the help of a protein complex called FACT. FACT stands for facilitating chromatin transcription. This protein complex removes a histone dimer H2A, H2B from the nucleosome, leaving behind a histone hexasome. After polymerase II has moved past the histone hexasome, fat and other accessory factors help to deposit the histone dimer, thereby restoring the structure of nucleosome. Elongation takes place by the continuous addition of ribonucleotides to the 3' hydroxyl group of the RNA chain. Like initiation, it also takes place within the transcription bubble. In both initiation and elongation, incoming ribonucleotides that arrive at the transcription bubble are complementary to the corresponding nucleotides of the template strand. For the addition of each nucleotide, a phosphodiester bond is formed between two adjacent nucleotides. In case of error, RNA polymerase rectify by proofreading. During elongation, in addition to RNA synthesis, polymerase performs an impressive range of tasks. Number 1, it unwinds the DNA double helix in front and rewinds it behind. It also dissociates the growing RNA chain from the template strand as it moves along. And finally, it also performs proofreading. During transcription at any given time, only 8 or 9 nucleotides of the RNA chain are base paired to the template strand. The remainder is peeled up and projected out of the enzyme through the RNA exit channel. In this way, the RNA chain is extended. The last stage of transcription is termination. In eukaryotes, there are two types of termination. One is factor-dependent termination and another is factor-independent termination. At this stage, the transcription complex is dissociated and the RNA transcript is released. In addition, the final RNA processing event, that is 3' polyadenylation, is also coupled to the termination of transcription. Friends, this is all about the mechanism of transcription in eukaryotes. For more updates, subscribe my YouTube channel, Sam Biology Class.